10 meter step 135.
Looks all stable when we zoom out.
It's deceiving, isn't it?
What about the
It's a long
single
way in there.
branches?
Those are either primnoids or bamboos. We've seen a million of these today.
Yeah.
They look like they're primnoids.
Okay.
And
Roger. It's a nice Argus shot, though.
We have some viewers wondering why Argus is bobbing a lot more today than in some of our other dives.
it's because the ship is bobbing a lot more today.
Yep. So Argus moves with the ship. So it's a critical component to these expeditions because Argus takes away the movement from the ship. So Herc can be a bit more nimble and kind of fly around the seafloor a bit easier than also bobbing with the ship.
We're at 1705, so we're still getting some of these spectacular animals here. I'm not sure we're going to find out what their upper limit is today because we're going to be heading up in about an hour or so.
Yeah.
But we know what it isn't.
Yep.
That's half the battle.
Yep.
For everyone watching too, this dive will be wrapping up in just a few hours. We hope to be back on deck around 8 Hawaiian time. And then we're going to transit south and map a bit to get out of some of these big swells that we're experiencing. So hopefully some nicer weather, a bit more south in the monument.
Yeah, we're going to map until we nap. I love mapping, believe it or not. I've done a lot of it over the years, and it is just another form of exploration. You know, we'll get sort of a lumpy sort of satellite image of a seamount, but we really don't know what it looks like until we map it. And when we map it, we're revealing the actual topography of the seamount. And I get just as excited seeing some of these seamounts as I do these corals and things.
Are you hovering?
It's the same thing.
Mm-hmm.
It's all exploration, all seeing things for the very first time.
Look at that. There's no wrap in your tether. We just moved, so we're...
Might be a fish in the water column over there in the left quadrant, upper quadrant. I don't know if he's going away from us.
Left quadrant. I'm going to let Antonella back in here now that I've put a turn in the tether. Thanks, everyone.
Okay, thank you, Dan.
Thanks, Dan.
Happy to be of service. Once we're settled in, are you guys ready to keep going?
Yes, please.
Okay. Let's get back on the 131 and continue our way back up to the top. Lincoln. All right, ready to go.
So
Bridge nav, 20 meter step 131.
So Aaron, can you tell me about how much longer we've got in this dive?
Well, Trevor was estimating about 2 hours. Are we certain on that, or should we be going a little earlier? Okay.
We're uncertain on that. Let's see. So how deep are we?
1,700.
Yeah.
What rate are you getting these days with all those
About
rocks?
15, which puts us at 113 minutes, just quicker than 2 hours.
Okay.
So I would say like maybe 5 minutes before the top of the hour we could leave to be a little conservative.
All right, that sounds good. Yeah, I'd like to be on at 8 on deck as the board instructs us to do.
Raj.
So then we have about 35 more minutes, if I understood that correctly. Is that about
25,
right?
yeah.
25, okay. Oh, right. I see it now. It's a good thing I didn't stick with math, I guess. Started out as a math guy in college. Let's see. Sorry, I'm just catching up with chat here from our science chat line. Let's see. I'll answer Ken Sulak's question first. He's one of our fish experts. And yes, Ken, I was referring to the OER guide in the photo gallery. It's going to be in the Scorpeniform group in that guide. And there's only one image, and he's kind of on one of the first rows of that guide. If you want to go try to look at them.
Meters 131.
And then Asako Matsumoto is talking about the scleractinian. And let's see what she's saying. She was talking about also Dr. Stephen Cairns with the Smithsonian. And he only uses the skeletal information for the identification of scleractinians. And of course, the whole taxonomic field is moving slowly toward more genetic identifications. Yeah, so Stephen Cairns is known to quite a lot of us, as he is one of the premier deep sea coral taxonomists. And he's also a fabulous person too.
You must be excited to see that sponge.
Oh yeah, yeah. I'm always excited, and I'm also excited to see corals too.
But that sponge you hadn't seen before?
Oh, that one you're talking about.
Yeah.
Oh, what, you could tell I was excited?
So what's the process that it's going to go through when it comes to shore? So what will you do with it?
Well, the first thing Steve Oskovich is going to be down there prepping the lab for, he knows what all the specimens are, including the rocks, and he's going to get everything all set and everything sort of ready, labels, pans, his data sheets, and so forth. And you can actually watch that process if you want to, because it's filmed and there is a camera in the lab itself. So the ROV will come up, the pilots and everybody will get it ready. We don't immediately go running to the ROV. They've got to do some things to make sure it's safe and everything's secure. And then they'll give us signals when it's safe to approach the boxes, and they'll open up the boxes. And then we'll go there and we'll pull these specimens out of the boxes as carefully as we can.
It's like trying to keep dogs away from their bowl of kibble. So eager to get at it. Yeah, stay back, stay back.
I was thinking if we got enough scientists, we could mob you guys in sort of like a scientist pack, sort of a wolf pack.
We have some defensive maneuvers, which are using the ROVs in their dangerous ways.
The blinding lights.
Well, I was sort of thinking they're more of an electric fence, given the electricity that goes through that thing. But anyway, yeah, we don't want to touch it until they're ready for us to touch it.
If you're watching and you look at the quad, it'll be in the bottom left corner, the wet lab.
Yeah, and then once it gets into the lab, they first go into a refrigerator to keep them a little bit chilled before we can get to them. A couple of rocks Dr. Orcutt will begin with immediately. These are the rocks that she's going to try to work with microbes out of, and the other rocks are going to be just set down and dried out, and we'll deal with them a little bit later. And then we will pull each of the biologic specimens out one at a time, and they're placed in front of a screen with their ID numbers and a measuring scale, and they're photographed very, very carefully by Steve. And then once he's done photographing them, then we'll decide what type of container
20 meters 131.
we're going to place them in, and we'll put those in that container. It's usually 70% alcohol, and that's what we're going to do with the sponge, is put them in 70% alcohol.
Oh, what is that thing right there?
That's another one of these. We can skip this. This is one of those I4 beautiful candelabra that they were just imaging while we were eating dinner. Again, and they've done this twice.
Okay.
Anyway, so he will photograph them all. We'll get them in the containers, and they'll be put back into the refrigerator, and we'll go to the next one.
And so how long will it take your team to figure out if it is a new species or a species that hasn't been to this area before?
Well, I'm going to take a small piece here on the lab. The bulk of the specimen will go to the repository on the East Coast. And I'll take the small piece, and I can tell you that I'm going to look at it as soon as I get home, because I have a little basement lab. I'm afraid I'm that much of a nerd that I've got my own basement lab. And I will photograph. I have a camera set up on my microscopes there, and I will bleach out all of the spicules, and I will photograph them very carefully, and then I will clean all the photographs up and put them on what I call a spicule sheet, and then I will send that sheet and all the information, in situ information, to a colleague in Moscow, Dr. Konstantin Tabachnik, and get his opinion on what this is. I'll, of course, try to research it myself and come up with my guess, and then I'll send it to him and get his thoughts on the sponge. And he's much more of an authority and specialist than I am, and when he comes back with his ID, then we'll kind of know whether it's brand new or whether he's seen it before. And that's the process.
Very cool. Thank you.
Yep.
This is cool looking.
I don't know. I think Sponge Bob has some competition. There's Sponge Chris.
It's like a stick fellow.
Oh, look at all those stars just kind of falling off.
Yeah, those are brittle stars. They bail
Thanks,
like that.
Antonella.
Falling stars. And
And the top guys are feather stars. They're actually in the crinoids. It's a class of the phylum Echinodermata.
so why did
That's
they bail?
pretty cool.
I don't really know. It's some
Pictures.
response reaction, I think, to the lights or the water coming from the ROV thrusters, or
They're just trying to show off.
Nice circle, Antonella.
Mhm. And what's the stalk?
the
Is
stock
that a
is,
different?
yeah, it's a dead sponge, and on the very top of it are these yellow sea lilies. These are actually in the family Chiritometridae, and most likely they're in the genus Glycometra. These are fairly common here at these depths. Beautiful feather stars, but we have sampled them before, and we've sent them to be looked at by people who specialize in that group.
So you wouldn't want to zoom on them.
Bridge nav, 20
Did all
meters
of those
131.
sponges,
Can we zoom on them?
stars,
Cool.
kill the sponge?
Can you get
No, probably not. The sponge just died, and the brittle stars then
Right.
decided to crawl up there and use it as a perch.
Viewer wants to know why sponges and corals are
It's okay on this one, 'cause we're a
animals
bit far
rather
now, but
than plants.
Excuse me, what was that?
Why corals and sponges are considered animals rather than plants.
Oh, because they're not plants, they're animals. So the biology is pretty clear cut. You know, plants survive and they acquire energy through photosynthesis, and these animals do not. However, these actually acquire organic material from the water column itself. The cells are animal cells. They don't have
get ready to take pictures on Argus.
don't have any of the chloroblasts and chlorophyll and everything else you need to actually use sunlight as an energy source. So, nope.
Are you trying to land Argus? Is
They're
that what
animals.
you're aiming for?
Thank you.
I just trusted. I know you know what you're doing, Trevor. I'm just joking.
couple of viewers want to know if we have come across any man-made items.
You know, normally I would say no, but we suspect we have. And what I mean by that is several dives ago, and I was not on this watch at the time, they came across a rock that did not belong. It clearly did not belong where it was. Totally different color. It looked like it had carved faces and a ridge on it. That was sent both to some specialists in Hawaiian culture and Hawaiian, how should I say, Hawaiian artifacts. It was sent to a geologist as well. And the conclusion was that it is a man-made rock, and it was most likely a very, very early form of anchor or ballast stone that somehow was left on the seafloor way out here. Hopefully it wasn't a tragedy where some canoe that was trying to make it to Hawaii overturned and this rock went down, but there's no way you're ever going to know the story of why that rock was there.
So there may or may not be an ROV pilot that is not on board right now and shall remain nameless, that when a hidden rock comes up on the ROV and is not needed for science, as in they don't know where it came from, so cannot use it for a scientific sample, this aforementioned ROV pilot will keep that rock for a little while, and then as soon as the ship's traveled a significant enough distance, we'll throw it overboard in hopes to confuse future seagoing operations.
That would have been a trick. This is a pretty big rock.
Bridge nav.
I mean, if it was used as an anchor, which we suspect it was, I actually looked up forms of very, very early, they call them weight anchors, and this is before they really understood that you've got to grapple, put some fins or something on it, wooden fins and things to grapple the substrate. They were just trying to use this anchor as simply a weight to hold the boat in place. And I suspect it could have been something like that.
Or our maritime archaeologist, Hans von Tilburg, his best guess is that it was a weight for some kind of open ocean fishing gear. Thinks it's too small for an anchor, and he thinks that nobody would actually throw ballast overboard at that open ocean area. So that's kind of what his ideas were.
It looked big, but I guess I didn't really see an image with the lasers on it. I just assumed it was big from the image.
Yeah, it wasn't that large. It was.
Oh, okay. A fishing weight, okay. But it was man-made. I think he believes it is too.
So it's about 20 centimeters at best.
Centimeters. Okay, I didn't even notice those dots right on the top of it. So yeah, I guess that would be too small to serve as an anchor. So open ocean fishing weight?
Yeah, a weight for open ocean fishing gear is one suggestion he had. I mean, maybe not
That's
current,
a really long one.
Not current gear, but older gear.
You're looking at an unbranched bamboo.
Beautiful.
That probably reaches the surface. No, I guess it doesn't.
Are you happy for me to zoom in to know?
That's a long one.
Dense. Those polyps are quite dense on that.
Bamboo coral in the family Keratoisididae, excuse me.
And the bamboo comes from the keratin kind of this separations in the structure, the stem structure.
You want to end this beautifully? Let it leave the frame.
Which you can't see on this one so clearly, but you can see on others very clearly.
Yeah.
Beautiful.
Nice.
Nice job there, Antonella.
That was awesome.
Yeah. Nice work.
Well done. That was nice. Is that a fish? Or is it just a black line? I think it's a black line.
That's what's referred to as self-correction.
I'm doing it all the time.
Bridge
I'm just
nav.
muted.
I know, I do.
20 meters 131.
I do it all the time too.
So another viewer is wondering
All right,
if
you're going to want to make it over. Sorry.
Go ahead.
Sorry, what was that after data?
Yeah, so if you've seen any major morphological differences between separate populations of a species of sponge or coral at the different seamounts.
That's a good question again. Boy, oh boy, I hope the other watch leads get these type of questions. I'll tell you.
I love these questions. Please keep them coming.
So the answer is yes. I think the first seamount that we visited did not have any of these sponges. They had a completely different batch of sponges that we consider to be in the family Farreidae, and we actually sampled one of those sponges, but none of these were here. And on this seamount, we've seen hardly any of those. And let's see, I'm trying to think of the second seamount we were at. I'm pretty sure there were none of these either on that one, but then it had some different ones from the first one. So you just never know where you're going to get when you open a box of seamounts.
Yeah, and that's
box of seamounts.
Don't you get that?
I get it.
Tom Hanks, right?
I get
Lots
it.
of chocolates.
Yes.
All right, never mind. It was a stupid joke.
The scale was a little off.
Yeah, a little bit. But anyway, very good question.
Yeah, thank you for that question. Here, a question for the pilots. This viewer has heard that Herc is very loud when in motion. Would you please provide a comparable land-based vehicle sound? This is from Patricia. Oh, I don't know where she is.
I think it's going to take a minute. They're working on something.
Okay. Thank you,
Well,
Erin.
let's go to another one and come back to that one. You got another one?
Yeah, you know, a couple of viewers have said that the second dive of this cruise found a 1968 7-Up can.
Is that right? It was not on.
We've seen about 2 soda cans.
He was just
I didn't see that. It wasn't on my watch.
He just had Argus down looking like great.
I believe him. I haven't, it wasn't on my watch and I didn't hear about that.
The second dive.
Oh, okay. That would have been on hhmm seamount on the eastern side of hhmm seamount.
Yeah, and would you tell our viewers why we're not seeing this human-made items?
Well, we're pretty far away from any type of human settlement or city or town or anything like that. So, you know, any type of human debris like that would have to travel quite a ways, and then it also has to get down about 2,000 meters. And a lot of human debris actually floats closer to the surface, so it doesn't make it down here. So that's one of the reasons. But whatever the reason is, I sure am happy that we don't see a lot of them.
So Trevor and Antonella, our pilots, can you guys see if you can answer this question from one of our viewers?
Hit me with a question.
So this viewer has heard that Herc is very loud when in motion. Would you please provide a comparable land-based vehicle sound?
Land-based vehicle sound?
Yeah.
I don't think we get to have a land-based vehicle sound, but what's a good land-based sound that's comparable to Hercules? That's a good question.
Rat tails.
Would it be like the noise right before we go versus the high voltage and the thrusters?
Yeah.
It's noisy for sure.
Yeah, what does that sound like? I mean, it sounds like Herc. I don't know.
Yeah, like a screeching Herc.
For the industrial viewers, we could say it sounds like a hydraulic power pack or HPU, hydraulic power unit. That probably doesn't explain things to 97.4% of the viewers.
Sorry, is that starfish eating coral?
Yes.
Oh.
Off to the right?
Yep.
It's almost done with it too.
Oh my gosh,
Oh,
is
no,
that the
still got
evil star?
It could be, or it could be another one too.
Maybe a really loud, aggressive vacuum?
Yeah.
Oh.
An old one that's like plugged in and the wrong RPMs or something.
Right, yeah.
That's kind of what it sounds like. Sounds like when the crew gave me a shot vac and told me I could use it in the sonar room and it was not the right
Voltage.
Yeah.
Start smoking.
I was screaming, and I told them, they're like, oh yeah, we do that all the time. I'm like,
That's
I don't think
awesome.
that's a good idea.
That's why our vacuums keep getting broken.
That's right.
We went to battery operated for that reason.
Wow.
Oh, a bamboo coral. It's eaten most of it.
I wonder how long it took it to
Look at that guy. Just lounging.
Yep, eating about half of it, not most of it.
Fat and happy.
But here you can see the nodes in this bamboo coral, so that's one nice feature to be able to see those clearly.
So you're saying the sea star was justified in eating this coral so we could
Yes.
see the nodes?
Yeah, the sea star is giving us a demonstration.
I see. Okay.
Look at his balance
So what
there. He's so
kind of star
precariously
is
like
this?
balanced. It's a tightrope.
Really like that Argus view from the side. It's cool.
I am attempting to identify this sea star. It's not Hipposteria. It may be.
That's all I have for Zoom.
Okay, thank you.
Do you want me to pull out for you to? Okay. Yep, we're quite a ways away.
I think this guy is called Evoplosoma. Yes,
Ooh,
you were
they love
asking
the
about
evil,
that.
Yeah.
Mm.
Only got time for a couple more questions if we've got any more, because we're very close to ending the dive. Well, actually, we do have more
I was gonna
question
say,
time. We just
we
don't
have
have
a lot
much
of blue
bottom
water
time.
for it.
Yeah, that's right. So it's just we won't be seeing the seafloor for the remaining part of the dive in about 5 minutes.
Oh,
Those barnacles. Look
already?
at all those barnacles.
Did we see any jellyfish in this dive?
We did at the end of the last dive. They saw a spectacular one.
The viewer wants to know about Halitrephes maaceae.
About what? Can you show it to me?
Halitrephes maaceae, jellyfish.
I have no idea. I don't know. I don't know my jellyfish, so I'm sorry, and I don't know that particular one. Oh.
Well, we made it almost to the top, Chris. We did pretty good
Yeah.
on this watch.
Yeah, this is great. Thank you. You're actually at the top pretty much, because
Yeah.
you're in the final contour right now.
Yeah, we are. But that's a big final contour. It's like 40 meters to get across it. But I think we done well.
Yeah, and we do know that there are still members of that community that persist up here, but it's a much more sparse grouping.
Are you getting some pictures?
Yeah, I got you.
Yeah, thank you.
Yeah.
My high school students in Los Angeles would love to see those
This is really
pictures. Thank you.
So one thing this shows is the colonial nature of the corals, that it doesn't depend on, so the corals to the right that aren't consumed yet are still alive and feeding. And of course the ones to the left are gone, but they're able to survive without the entire thing being alive.
Oh, and they're just waiting there to be.
Yeah, it's kind of a desperate, horrific, slow motion scene.
We have another complex question.
Are we supposed to do something at the end of the
Time to go.
Yes.
dive? Is that right?
Oh yeah, we need to do our protocol.
So we're going to pause for a moment before we take more questions.
So we are very grateful to this place, Papahanaumoku Akea Marine National Sanctuary. We have
Monument.
Oh, I'm sorry.
Not a sanctuary yet.
Not yet. Monument, I'm sorry. And we are very happy to have seen such great biodiversity of sponges and corals and fish and other organisms. So at this moment, we are going to go off the air to conduct our private ceremonial protocol to acknowledge and honor the biocultural resources that are present and that we are interacting with and impacting. So we will be back shortly on SPL.
Okay.
And then, sorry, we have a request. If we can get a Niskin sample when arc is between
Oh, yeah.
10 meters and 15 meters in altitude. So yeah, like between 10 and 15.
10
Sorry,
and
how many meters? 10 meters?
10
yeah,
to
10 it works. Yeah,
10 to,
just
10
says
to 15?
15.
Okay, sounds
Yeah.
good. We'll get set up for recovery first, and then we'll take those Niskins
Okay, thank
before
you.
we leave bottom.
good job remembering.
Thank you guys.
Absolutely, thank God for you remembering. I completely forgot about that.
Oh, that's my job.
I didn't even know about
The
it at
least
all.
I can do.
Oh, but there's so many things that are my job that I very much forget all the time. So good job.
We have to apologize to our videographer, Aaron, for not really providing her for many things to look at today and videotape.
Oh, but she had fun this
I
morning.
appreciate it.
That
We had
was
some fun fish.
that was being sarcastic. I think you're almost overwhelmed with the amount of material to see.
Oh, I love it. I love a good zoom, if you can't tell.
But you know, the community shots are pretty nice too.
Yeah,
Just
those were cool when you had, I like the Argus shot with everything in it.
Oh, especially the bolosoma sponges.
oh yeah, that yellow one was really
Oh my
beautiful.
God.
The one that, yeah, we saw a lot of things today. That's for sure. And a lot of fish. We had echinoderms, urchins, sea stars, tons of coral, tons of sponges, crabs.
It's easy to forget that was the same day as today.
I know. You know, I know exactly what you're talking
Yeah.
about because we were all on shift at 4 in the morning through 8 o'clock this morning earlier today. So this is our second shift.
Yeah, I'm really feeling Ola ke au a Kanaloa. Long live the waters of Kanaloa.
Mm-hmm.
Yep.
Hi.
Such beautiful life here in the deep sea.
So we're going to say goodbye to this site
That's
for
done.
now, but we are considering doing a second dive here. Just got to see how long this sea state is going to last and sort of do a little bit of planning. And then what we decide will be posted on social media, as Kelly mentioned earlier, when she was subbing for Aphrodite.
Bridge nav, we are getting ready to leave the seafloor. We anticipate about 2 hours coming up. We're hoping to be on deck close to 8 p.m.
And you have Niskin 5 open.
Sorry, Science Road, could please confirm 10 to 15 meters? That's 10 to 15.
Yes.
Thank you.
Thank you. And sorry, just make sure you have Niskin on 5.
Popped.
Way to go, Antonella. That was a tricky one.
What sample was that?
That was sample 087.
087, thanks.
Yeah, thank you guys.
All right. We are now in blue water, and so have we got some questions that we can try to answer.
Oh, yeah.
You're just bumped in now.
Oh, so that starfish, the Evoplosoma, is that an invasive species?
No, not at all. It naturally occurs here. It's not like the crown of thorns or anything near the surface. It's just in deep water. These sea stars are known predators of the corals. That's how they make a living, if you will.
And then when do you see most active sea life such as squid, sharks, and fish?
We saw a fair number of fish today, but we would potentially see more of them if we were diving on more sediment type of substrate, but we are diving on rocky substrate. There are not too many sharks that make it down this deep, but there are a few. We haven't seen them so far. The 6-gill sharks, we're a bit deep for 6-gill sharks. We are not too deep for what they call sleeper sharks, so but we haven't seen any. And squid are often further up in the water column, and we're usually down by the bottom, so we usually don't see very many of those. Occasionally we do. You might see some as we ascend for the next couple of hours.
Okay, and this is that other complex question. Can we do that now?
Okay, I'm ready for it. I'll do my best.
Why are there areas very densely populated with sponges and coral and others are bare? Is it related to current, food, temperature, salinity, gases, geology?
Well, that's the million-dollar question that we're trying to answer. I suspect current, and I suspect food. One thing, the geology, I assume, is in reference to the substrate type, and I think that's quite important. Don't know about temperature and salinity, but down here, those are pretty constant as you go from one location to the other, so I suspect that those aren't necessarily all that important from one area to the next. And then gases, I'm not sure which gases you would be referring to, but I think at least 3 of the things the viewer mentioned are probably part of the equation.
Well, thank you for the question and the answer, Chris. And more?
Huh?
Do you want more questions?
We got a bit of time to kill, and even if we see a midwater animal, I don't know what they are. So.
Okay, audience around the world, keep those questions coming. So back to the sea star, the Evoplosoma. Does it spend its life on the coral and then die when it has consumed the host, or does it move on?
Well, we don't really know because we think it takes that sea star quite a while to consume the coral, but we don't know. And the one we just saw, it consumed half of the coral. But does that take a day? Does that take a year? We think it's a slow process. And so that sea star may be on that coral for quite a bit longer. And then once it's cleaned off all of the polyps, I'm pretty sure he's just going to crawl back down the stem and try to find another one. But, you know, how long do sea stars live down here? No idea. Not a clue.
Yeah, we're trying to find that out.
Yep.
So a few viewers are wondering when our next dive is, and is there a dive schedule?
Well, as Kelly said
Oh,
earlier today, it's a little bit sketchy. One of the reasons we're not diving tomorrow is there is a sea state condition that the waves are going to be too high for us to dive. So what we're going to do is we're going to head south as soon as we get the ROV on deck. And there are some seamounts that we were planning to map before we dove on them that are further south and out of that weather system. So we're going to go ahead and transit south to be productive and do the mapping while we're waiting for the weather to clear up here. And as soon as it does, then we're going to come back up here and resume diving activities. The issue, as everybody knows, is you can't predict the weather. It could be sooner, it could be later. So you need to look at those social media posts in the Nautilus website, and they'll try to update that as quickly as they know.
Thank you, Chris, for that explanation. Going back to the bamboo coral, after the sea star eats it, does it regrow?
If the sea star eats the whole coral, then it's not going to regrow. If the sea star decides it's had enough and it crawls back down the stem, then I think potentially the coral can then cover up the skeleton that's been stripped of tissue. I think it's possible for the coral to sort of regrow and cover that again.
Yeah. Okay, we have a lot of questions here. Okay. Oh, all right. Well, I guess since I mentioned that I'm from Los Angeles, I'm a teacher, and I got this amazing opportunity to join EV Nautilus for 3 weeks. And I can't wait to connect with my students next week, and I will have some of the scientists here talking with my students. Maybe Chris will talk with the class, and Kainalu, maybe, our university intern.
I would love to.
I think what she's asking you is, what's your secret?
What's my secret?
Yeah, how did you get on this cruise? Because she, I think, is quite, or he, sorry. I think they might try to figure it out themselves, how they could score this position.
Yes, so you can go to nautiluslive.org and go to education. So these are, here we have different education opportunities. There are lots of resources, lesson plans ready to go. And under at sea programs, that's where there is a an application. I'm not sure if they're opening it yet because of COVID. We had to wait a little longer, but there are applications here for educators who want to be science communication fellows, and also applications for students, university students who can be interns. And right now you can take the opportunity to schedule a live ship-to-shore interaction where you connect your students through Zoom with the core of exploration here aboard EV Nautilus. So thank you for that question, and everybody is very excited to share their experience at different capacities here on EV Nautilus and just share their passion for the ocean.
And a lot of the people who are on the ship now started as interns. So it's a really great program.
And again, if you go to nautiluslive.org, you can click on team, and you will see every single person who is aboard EV Nautilus. There is their autobiography here. And they really tell a story about how they got to be on EV Nautilus. So thank you for that question. Oh, and this is a perfect question for our pilots.
Go for it, yeah.
Yeah. What do the ROV pilots do to keep their skills while in the off season, especially since there was a whole year off?
We mainly just come back and screw it up for a while. If I'm being perfectly honest, there's not really a good way to practice this except for being in it. We were actually talking about this yesterday, is how do you practice using the arm at depth? So it's not just using the controller to grab a rock or to take a coral sample or something. It's more than that. There's a more, sorry, hold on a sec. There's more complexity than just doing that. So Chris gave a great example yesterday of playing ping pong with one eye closed. You know, it's very hard to get depth perception without stereo vision like we're used to. And with these cameras, we only have one. So you think, okay, well, maybe I can practice on deck, right? Similar, same controller, same camera feed. But a big difference is there's a different configuration of lighting when you're on deck. You get the sun or you get the lights in the shop hangar. And it turns out lighting is a very big tell for us using the manipulator subsea. There's slight shadows. There's, of course, I want to say it's aberration. Is that the right word for when res disappear? Is that the right term? Anyway.
Attenuation.
Attenuation, yeah, where the color changes the farther away you get in water. So all those subtle cues are really big hints on depth perception, which you cannot mimic on the surface. So even if you had a full functioning ROV in your living room, which why would you have that? If you had that to practice in the off season, it still wouldn't quite simulate what you're doing subsea. So I say it jokingly, like, yeah, we just screwed up a bunch when we come back, but it's kind of true. We just have to keep doing it in situ in order to get and stay good at it.
And by the way, I think Antonella is working to become a pilot right now, and we've been watching her in this cruise, and like today, I saw obvious difference. You are getting that sense already, and it was a really good job today on there, and it's just, I've just noticed the sort of the growth and how you're getting all those skills.
Yeah, she's crushing it.
Thank you.
So you don't have a video game trainer that simulates a lot of that stuff, huh?
Okay, what else we got?
Oh, another question for the ROV pilots.
Okay.
Are you driving the ROVs to the surface, or are they pulled up by a cable?
Yeah, so Argus is being winched up by our big, well, I guess you probably don't see it on screen, but there's a big winch that it's attached to.
I can put it on
Herc
screen.
Is, oh yeah, that'd be cool.
Let me do that. So we'll put it on the quad.
Okay, so Aaron's gonna show you the winch, and that's what Argus is attached to. So it's winching up, and Herc is driving up with its thrusters. So basically, we're limited by Herc's speed on recovery because we've collected a lot of rocks and such, and so Herc can't always come up as quickly as the winch, and so we match we match the winch speed to Herc's speed as we ascend.
So if you look at the quad view, in the bottom left will be the winch. So that's the winch room.
There was a question I saw earlier about what was the chant that we were, the ceremony. I wonder if Kainalu could address that a little bit. So we haven't really put him on the spot very much today.
I think we sung, did we sing the chant when we enter the monument?
Yeah, just a little bit about what we do when we go silent for a few seconds.
Yeah, so yeah, those are what we call oli in Hawaiian, or mele is another term, also commonly known, as Chris said, as chants, but it's basically we set our intentions and ask for permission before conducting our work and as we enter the space. So yeah, these only were conducted, I believe, by the cultural working group, Andy, I want to say.
Yep, cultural working group, yes.
And so, yeah, it specifically was created for this type of work, for research in the monument. And we do one before we start our shift and begin our dives and collect samples. And then we also do one when we end our dives before we come back up as a sort of mahalo or thank you for the place and acknowledging our work in this area, as also like a release, or we call a punny or closing. So usually when we open, we also have a closing to something. And so, yeah, that's a little bit about our Oli, and the OAT team has been awesome at learning it and doing it each time. So I'm very appreciative of yeah, this Nautilus cruise and being so open to doing these Oli.
We've got some great teachers, very patient teachers as well.
Yeah, I mean, this isn't easy stuff, you know, the language is hard learning, so it's really about the intentions that we set. And it's been great. I've seen a lot of awesome things, so.
I gotta admit, I was pretty anxious before we actually did it the first time, but now I really like it.
Yeah,
I kind of look forward to it.
Honestly, I can hear you guys getting a lot better each time we do it more and more. And this is my first time coming out here and actually learning this as well, so it's been new for me, and it's a great one I'll add to my list of other Oli to offer when I do other type of research as well. So yeah, that's a little bit about our chance, I guess. Yeah, thank you for that question.
Yeah, thank you, Kainalo. So for our viewers too, if you are more interested in learning about the Hawaiian culture and especially the naming of the expeditions, you can go to nautiluslive.org, and if you scroll all the way down to the bottom, there are a few, there's one recent highlight. What's in an expedition name? Building relationships between people and place through Olelo Hawaii, the Hawaiian language. And maybe Andy, you can tell us a little more about the name of the expedition?
Sure.
Thank you.
Lu'aea Hiki'i ka Papaku, and it's more of a literal name of actually what we're doing here. It's symbolic. It basically says diving up and down and then doing work on the Papaku, which is the ocean surface, or the ocean, the bottom of the ocean. And so that's where the name for this expedition comes from, and that was given by the Cultural Working Group for Papahanaumokuakea, and they advise the managers of Papahanaumokuakea on a number of different things related to cultural integration and cultural appropriateness. You know, if you have many of the monument managers are federal agencies or state agencies, and they certainly don't have a lot of expertise in evaluating, say, like a project, a culturally based project, which is coming into this region. So having a cultural working group that's comprised of leaders in the Hawaiian community around the Hawaiian Islands as a consultation group, they can evaluate some of those applications for permits and provide valuable input on whether they are, you know, whether they have value or whether they really know what they're proposing. So it's been an incredibly valuable. There also the working group, as well as contingent of Hawaiian elders around the Hawaiian Islands, were really leading the original protections for this place. Some who have passed, unfortunately. Uncle Buzzy Agard comes to mind, And he was one of the leaders that went to Washington and urged for the protection of the northwestern Hawaiian Islands. And he was a commercial fisherman, you know, before that. And he had come up to this place many, many times in many years and saw how incredible, bountiful this place, but also saw how quickly it could be depleted. He was going to establish an Akuli fishery at French Frigate Shoals, but saw how quickly that resource could be depleted and changed his mind and thought that, wow, we need to protect this special, unique place. And we've had a few other Native Hawaiian leaders who have been involved in the expansion of the place, in our World Heritage nomination. So it's a huge part of our management, and we're incredibly grateful and proud and fortunate to have that. And it's also translated to other areas, how they work with their indigenous communities to share management, to enhance management of other areas. So, you know, we like to think that we've been kind of a leader in that effort, and this mission is an example of that. So, yeah, that's a kind of a long-winded explanation, but there's a lot of history here.
We got a lot of blue water to fill, so it's okay.
We do. Yes.
No, it's very special. I'm really appreciating the connection, the education, not just about science, but really the education and the cultural significance of this place. So, and there's a beautiful explanation there on our website. When I would like to read the Oli, the chant, because I think it's so beautiful. So if I may, and I please excuse my pronunciation if I make mistakes, but so it starts with Lu'u a ea, ahiki ike kai, lipo lipo, and that means dive and rise until reaching the deep blue ocean. The second line, Lu'u a ea, ahiki ika papaku e, dive and rise until reaching the seafloor. Lu'u a ea, ahiki ike ku alono akai, dive and rise until reaching the sea ridge. And the last sentence, Lu'u a ea, ahiki ike kumu e, dive and rise until reaching the foundation of the ocean islands. So that is so beautiful.
Yeah, and those are the names of the different expeditions, right? Yeah.
And so our viewers can go to this page, and there is this Oli is recorded, so you can hear it and sing along with it. So yeah, thank you for that question. And let's see if we have any other questions here. Okay, so it sounds like many of our viewers really enjoy this dive, this 13-hour dive. Some of them watched the whole thing.
Wow, that's fantastic. Yeah, it was a very, very special dive for sure. I mean, we saw some incredible coral and sponge communities. We saw a lot of animals that we hadn't seen on these dives before. The chimera fish was the one that comes to mind, beautiful jellyfish, as well as some urchins, other fish. It was a very vibrant and abundant community that we saw, a lot of diversity. And we collected a lot of fantastic video and imagery from this. I mean, I was blown away. And as we mentioned before, Chris, who has done many of these dives over the last 20 or so years, you know, feels like it ranks up there in his top 5 of high density communities and these unique communities that he's seen in the past. So I think that says a lot.
Yeah.
I wonder what people are doing when they watch it for 13 hours straight.
What would
I
you
remember
do?
we used to have like a knitting group that used to watch it.
Oh. What would you do, Aaron?
That's a lot of hours to watch it.